Hello everyone, I'm Dr. Indira from the Department of Life Sciences, Kristu Jayanti College. In my lecture today, I shall be talking about a bryophyte called Riccia. We shall be discussing about its structure and reproduction. As we all know that bryophytes are commonly called as amphibians of plant kingdom, Riccia is a commonly seen bryophyte growing in the marshy or wet soils. It is classified under the class Hepaticopsida, order Marcantiales, family Ricciaceae. There are many species of Riccia which are cosmopolitan in distribution. There are over 138 species of Riccia which are found distributed in both the tropical and temperate regions of the world. While most of the species are terrestrial in habitat, Riccia fluitans is an aquatic species which is found floating in ponds and lakes. The most common species of Riccia which are found in India are Riccia discolor, Riccia gangetica, Riccia frostii, and so on. The name Riccia comes from the Italian botanist Ricciai. In this slide, let us look at the structure of the thallus. The thallus of Riccia is dorsi-ventrally flattened. It has a dorsal as well as a ventral surface. It lies flat on the substratum. The plant grows by dichotomous branching. There is a growing point situated at the apical notch. On the dorsal surface are seen a prominent, well-defined midrib, while on the ventral surface are seen a number of rhizoids and scales. The rhizoids are of two types, namely smooth as well as tuberculate. The smooth rhizoids are meant for absorption, while the tuberculate or pegged rhizoids are meant for support. Along with the rhizoids are also seen the scales, which are simple, ligulated, and are meant for protection. They are multicellular in nature and about one cell in thickness. The internal structure of Riccia thallus is differentiated into an upper photosynthetic zone and a lower storage zone. The photosynthetic zone comprises of compactly arranged vertical rows of chlorenchymatous cells, which are separated by air chambers. These chlorenchymatous cells contain chloroplasts and hence are involved in photosynthesis. The storage region lies below the photosynthetic zone, made up of compactly arranged parenchyma cells and are filled with starch grains. Few cells of the lower epidermis of the storage region produce rhizoids. Riccia reproduces both vegetatively as well as sexually. The vegetative reproduction is brought about by a number of ways. Death and decay of the older portion of the thallus, where the older and mature parent thallus dies and decays. Growth is brought about by the apical notches. By adventitious branches. The adventitious branches develop from the ventral surface of the thallus. These branches develop into new thallus. By persistent episis. Due to extreme conditions of summer or heat, the entire thallus of some species dries and gets destroyed, except the growing regions. Later, it grows deep into the soil and becomes thick. On the onset of favorable conditions, a new thallus develops. This is more a method of perennation rather than multiplication. Tubers are another method of vegetative reproduction where the thallus gets swollen up or thickened on the margins. This happens during the unfavorable conditions. On the onset of favorable conditions, these tubers develop into new thalli. In some species of Riccia, the apical portion of the young rhizoids divide or redivide to form gemma-like mass of cells. These cells contain chloroplasts and are capable of developing into new thallus. The sexual reproduction in Riccia is of oogamous type. The male reproductive structures are called antheridia, which are borne in the antheridial chambers, while the female are called as archegonia, borne in the archegonial chamber. Most of the species of Riccia are monoecious, while some are dioecious. The sex organs arise in acropetal succession. The antheridium, which is also called as the male reproductive sex organ, is differentiated into a lower short stalk and an upper body. The body of the antheridium comprises of a single layered jacket which encloses numerous mass of androcytes. Each androcyte differentiates to produce a single biflagellated antherozoid. At maturity, in presence of water, the antheridial jacket breaks open and these androcytes or the antherozoids are liberated out. The liberated antherozoids swim in a thin film of water or rain or dew. The archegonium is a flask-shaped structure differentiated into a narrow neck and a lower bulbous venter. The neck encloses the neck canal cells, while the venter encloses a venter canal cell and a large egg cell. For fertilization to take place, water is very important. Just before fertilization, the venter canal cell and the neck canal cells become disintegrated, forming a mucilaginous mass which comes out through the neck of the archegonium. This mucilaginous mass is chemotactic in nature, hence the antherozoids are drawn towards it. One of the antherozoid travels or passes through the neck of the archegonium, comes in contact with the egg cell and fuses with it, forming a diploid zygote. This zygote is the first cell of the sporophytic generation and is diploid in nature. The diploid zygote of Riccia develops into a proper sporophyte and is deep-seated inside the gametophytic tissue. The sporophyte is differentiated into a spore sac or a capsule, while the foot and the seta are absent. The capsule comprises of a mass of spores enclosed within the outer layer of calyptera. The calyptra is a part of the gametophyte. Inside the spore sac are seen the spore mother cells, which undergo meiosis to produce tetrad of haploid spores. These spores consist of a mass of cytoplasm and is differentiated into an outer exine and inner intine. The exine is hard, thick, while the intine is thin and translucent. The spores come in contact with soil and are dispersed by the wind or rain. After the spores are dispersed, these spores, in presence of light, suitable temperature and moisture, germinate immediately. They germinate, producing a germ tube. A dense protoplasm then flows through the germ tube to its distal end, which cuts off a large terminal cell. First, a rhizoid is formed near the base of the tube. The terminal cell divides, resulting in the formation of two tiers of 4 cells each. Further growth results in the formation of a young flat thallus. In this slide, you can see the life cycle of Riccia. Riccia shows a diplohaplontic life cycle where the gametophyte and the sporophyte are dissimilar, and hence it is called a heteromorphic type of life cycle. I hope in this video you got enough information about Riccia, its structure and reproduction. Thank you for watching.